thanks for joining us for another fascinating journey through the world of science here on the Science Channel podcast. If you're enjoying getting these complex topics broken down, please do us a favor and give us a like and subscribe. It really helps the channel out.
Today, we're diving deep into a seemingly simple question, but one with surprisingly complex answers. And it really is, how do our cells manage to juggle all of their essential tasks?
Yeah.
Specifically, we're going to be looking at the mitochondria.
Okay.
Those incredible powerhouses within our cells and how they handle the dual demands of generating energy, ATP, but also at the same time, producing the crucial building blocks that are needed for growth.
Right, because you would think it's like trying to run a power plant in a manufacturing facility at the same time using some of the same core components.
Yeah, you would think that trying to do both at the same time would lead to some serious bottlenecks, right? I mean, one process is about breaking things down to fuel activity, while the other is about building new molecules. So how do they do both?
Right.
It's a fundamental challenge for all life, and how cells solve this has huge implications for understanding diseases like cancer, where those growth signals just go haywire.
Absolutely. And what's truly remarkable is that recent research has revealed a very clever and unexpected way that mitochondria tackle this balancing act.
Wow.
And our focus today is going to be on this new understanding, particularly looking at a fascinating enzyme called pyrroline 5-carboxylate synthase, or P5CS.
P5CS. Okay, I got it.
Yeah.
So for you, the listener, our deep dive today aims to uncover how mitochondria create these almost separate operational zones within themselves to manage both energy production and the creation of these essential building blocks so that they don't just have a complete metabolic meltdown.
Right.
It's complicated.
It is complicated. So let's start with the basics.
Okay.
Mitochondria are indeed the primary sites of ATP production through a process called oxidative phosphorylation, or OXPHOS, but you could just think of it as their main gig, their power generating mode. Right, the energy factories of the cell. Classic.
Yeah, right, exactly.
We all learned that one.
But that's not the whole story.
Okay.
Mitochondria are surprisingly versatile.
Oh.
They also play a vital role in making essential building blocks, particularly certain amino acids. For example, they're crucial for what's called reductive synthesis, essentially the process of building up larger molecules from smaller ones.
Interesting.
Of amino acids like proline and ornithine. And this happens within the mitochondria.
So reductive synthesis, meaning not just breaking down the oxidation, but also building up reduction.
Exactly.
Okay. And you mentioned that they use some of the same resources for both of those processes.
Precisely. Both OXPHOS and this reductive synthesis of proline and ornithine rely, in part, on the same precursor molecule, which is glutamate.
Okay.
Now, P5CS, this is the key enzyme that we're going to be talking about today, is the key enzyme that controls the rate of this proline and ornithine production inside the mitochondria.
I see.
So it's sitting right at a metabolic crossroads, competing for the same starting material that the energy-producing TCA cycle also needs.
Mm-hmm.
It's like two major production lines vying for the same critical raw material.
So how do cells avoid this tug of war? Do they just produce a ton more glutamate? Like, is that how they get around it?
That's a very logical thought.
Okay.
But the recent findings actually point to a much more elegant solution. What researchers observed is that when cells are forced to rely more heavily on OXPHOS, for instance, by growing them in environments where glucose is scarce, and they have to use something like lactose for fuel, P5CS doesn't simply get shut down due to lack of glutamate.
Oh, okay.
Which is what you might think would happen.
Yeah, exactly.
But that's not what they see.
So what happens instead?
Instead, it undergoes a fascinating segregation.
Okay.
The P5CS activity seems to relocate to a specific subset of the mitochondria within the cell.
It's like it knows.
It's like it knows.
It's like, hey, we're going to go over here.
Right.
And you guys, you deal with that.
Exactly. Not all of them, just a portion.
Wow.
It's as if certain mitochondria are specializing in building while others are focusing on energy.
Wow. So mitochondria within the same cell having different specializations, that's new to me.
Yeah.
How does that division of labor even occur?
Here's where it gets really interesting.
Okay, good.
It turns out that P5CS has an inherent ability to self-assemble into these long, thread-like structures called filaments.
Interesting.
And what the research showed is that when the demand for OXPHOS increases, this filament formation is actually enhanced.
Okay.
These filaments then cluster together, and as we noted, they're not found throughout the entire mitochondrial network, but rather concentrated in specific mitochondria.
So it's like P5CS forms these little construction crews in certain mitochondria, and they're like, all right, we're going over here, we're going to build.
That's a great analogy.
But how does physically grouping the builders together solve that resource competition with the energy producers? Like, I'm not getting that.
That is the crucial link.
Okay.
The mitochondria that are rich in these P5CS filaments share a very distinctive feature.
Okay.
They largely lack the internal folds called cristae.
Cristae. So those are the highly folded inner membranes where most of that ATP producing machinery resides, right? Like the ATP synthase.
Exactly. The cristaes are packed with the components of oxidative phosphorylation, including ATP synthase, the enzyme that actually churns out ATP.
Okay.
So picture this. You have some mitochondria that appear relatively smooth on the inside, and they're filled with these P5CS filaments. But then you have other mitochondria within the same cell that exhibit those characteristic folded structures, teeming with ATP synthase.
Oh, wow.
It's a visual manifestation of this metabolic division of labor.
That's so cool.
Specialized zones for building versus energy production.
So it's like one set of mitochondria prioritizes making building blocks, and the other is all about generating power. But mitochondria aren't static, right? They're constantly moving around, they're interacting, they're like little beings in there.
Yeah.
So how does the cell maintain the separation?
You're spot on. Mitochondria are incredibly dynamic.
Yeah.
They're constantly fusing together, splitting apart.
Yeah.
It's a process collectively known as mitochondrial dynamics.
Yeah.
And this research underscores that this ongoing cycle of fusion, the joining, and fission, the splitting, is essential for maintaining this segregation of P5CS containing mitochondria from the ATP synthase-rich ones.
So these dynamic processes are actively involved in this specialization. Like, how did the researchers even figure out that that was their role?
They used these very clever genetic manipulations to disrupt these processes.
Okay.
For instance, they examined cells where the genes for proteins called mitofusins 1 and 2, or MFN12, were removed. Now, these mitofusins are key players in mitochondrial fusion. What they observed was that without proper fusion, the P5CS containing mitochondria couldn't effectively separate from those loaded with ATP synthase.
Yeah, okay.
And consequently, the cells struggled to boost OXPHOS, even when energy demands were high. It's like the construction crew couldn't set up its dedicated workshop.
Right.
And it led to this overall inefficiency.
Yeah. That makes sense. If they can't come together and then perhaps differentiate, they're stuck trying to do both jobs in the same space. So what about fission, the splitting of mitochondria?
Right. So they also disrupted fission by deleting the gene for a protein called DRP1.
Okay.
And this had a different outcome.
Okay.
In these cells, while they could still perform OXPHOS,
Okay.
their ability to produce enough proline was compromised.
Interesting.
And this had real consequences. For example, it impaired the production of collagen.
Oh, right.
Which is a vital structural protein that relies heavily on proline.
Collagen needs a lot of proline, doesn't it?
Yeah.
So that's a tangible example of why this building block pathway is so critical.
Exactly.
So it sounds like both fusion and fission are necessary, but for different aspects of this balancing act. So fusion helps establish those specialized P5CS-rich mitochondria, and then fission ensures that there are enough of these specialized mitochondria to produce the necessary building blocks without compromising the cell's overall energy production.
Exactly.
It's like a coordinated cellular ballet.
That's a great way to put it.
It's amazing. It is a ballet, isn't it? But, I mean, this is all fascinating in cell cultures, but the big question is, I mean, does this even happen in living organisms? I mean, is this just a lab phenomenon, or is this actually happening in our bodies and animals and plants?
That is the big question.
Right.
And the researchers found compelling evidence that this mechanism is indeed at play in vivo.
Oh, wow.
They looked at tissue samples from human pancreatic ductal adenocarcinoma.
Okay.
A type of cancer.
Okay.
And what they observed was the formation of these P5CS filaments within the mitochondria of the cancer cells. But here's the key. Within the tumors, but not in the normal pancreatic tissue nearby.
Oh. So cancer cells, which often face those very stressful conditions within a tumor, seem to be utilizing this strategy.
Exactly. Tumor environments can be nutrient poor and oxygen limited. And these are conditions that would likely increase the reliance on OXPHOS for survival.
Right.
So the presence of these specialized mitochondria in cancer cells suggests that this metabolic adaptation might be crucial for for their ability to thrive under such stress.
Wow.
This raises some very interesting questions about potentially targeting this mechanism in cancer therapy.
Yeah, wow, that's a powerful connection. Now, even though these P5CS containing mitochondria lack that ATP synthase, are they just metabolically dormant otherwise?
Not at all. Interestingly, they discovered that these P5CS-rich mitochondria still maintain an electron transport chain.
Okay.
So that's the initial steps of ATP production. They just don't have the final ATP synthase to actually complete the process.
Okay.
And remarkably, they maintain a higher mitochondrial membrane potential compared to their ATP-producing counterparts.
Higher membrane potential, but no ATP synthase to harness it. So it sounds counterintuitive.
It does, doesn't it?
It does, yeah.
The prevailing interpretation is that this higher membrane potential is actually fueling the reductive biosynthesis pathways that are occurring within those mitochondria.
I see.
A higher membrane potential is linked to a higher ratio of NADH to NAD plus BA, which creates this reducing environment. And that reducing environment is essential for building complex molecules.
Okay.
So the energy is being used differently in these specialized mitochondria.
Yeah.
Not for direct ATP synthesis, but to create the optimal biochemical conditions for producing those building blocks.
So the energy is being channeled towards creation instead of immediate power generation in those specific organelles. That's a really clever trade-off.
Precisely. And this brings us to, I think, the core insight from this research.
Okay.
The creation of these metabolically distinct mitochondrial subpopulations appears to be a remarkably elegant evolutionary solution. It's the cell's way of resolving this inherent conflict between energy production and the synthesis of essential building blocks.
Yeah.
By physically separating these processes into different mitochondrial compartments. One set can focus on power generation, while the other prioritizes the raw materials needed for growth and maintenance.
So the mitochondrion, which, we often think of as the singular entity,
Right.
can actually organize itself into specialized subunits within the same cell to handle these competing demands. It's not just one type of organelle. There are functionally distinct versions working in parallel.
Exactly. It's a beautiful illustration of cellular efficiency and adaptability. Through structural organization and the dynamic processes of fusion and fission, cells had evolved this sophisticated system to simultaneously meet their energy needs and their growth requirements within their mitochondrial network.
It really makes you wonder about other ways cells might be creating these kinds of internal specializations that we haven't yet discovered. So for you, the listener, think about this. If cancer cells rely on this specialized mitochondrial behavior,
Right.
could disrupting their ability to form these metabolic subpopulations be a potential therapeutic strategy?
That's a great question.
And more broadly, what other seemingly conflicting cellular processes might be resolved through similar forms of organelle specialization? I mean, who knows what else is going on in there?
Absolutely. I mean, these are crucial questions that highlight how much more there is to learn about the intricate workings of our cells.
This has been a truly deep dive into the inner life of our mitochondria. Thanks for illuminating this fascinating research.
My pleasure. It's always rewarding to uncover these surprising levels of organization within these very fundamental biological processes.
Absolutely. Well, as always, don't forget to like the video and subscribe if you haven't joined us yet.